Hi, everyone. I'm Dr. Sheila Segerson, the Director of Shelter Solutions at Maddie's Fund. Welcome to Maddie's Monthly Behavior Connection, a monthly webcast that's all about animal behavior. We are honored to welcome our guest today, Dr. Karen Overall. Dr. Overall is a professor of behavioral medicine at Atlantic Veterinary College, a University of Prince Edward Island, where she created the clinical didactic and research program, which trains veterinary students, residents, and graduate students. Dr. Overall has been named the North American Veterinary Conference Small Animal Speaker of the Year and was named one of the Bark Magazine's 100 Best and Brightest, Bark's list of the 100 most influential people in the dog world over the past 25 years. And she's certainly had a very big influence on my career, and I know many of you who are in the audience. So today, Dr. Overall will be talking to us about puppy socialization. Welcome, Dr. Overall.
Thank you. And whoever muted me while my rescue border collie was barking in the background, thank you at the beginning of the explanation. Yes, we're going to talk about socialization, and I just need to share my screen. And you can see that, yes, Sheila?
Yes, that looks perfect. We're good to go.
Perfect. Excellent. So we're going to talk about puppy socialization. And if my voice sounds a little croaky, it's because, yes, I've been at the gym a lot, and everybody there, as of 48 hours ago, seems to have developed this hideous cold. So we all sound awful. My apologies. Great. This would be great, except now I can't see my own. I'm back. There we go. Okay. So let's talk about what socialization means for puppies. And if I start to croak, I apologize. The first thing that we need to think about is neurodevelopment. And before I explain what that means, I need to tell you why I think this way. So many people in this field come from so many backgrounds. There are very few fields where you're going to see the diversity in training that you see in people who are committed to working with behavioral issues in dogs. But here, when people are committed to helping cats and dogs and horses, people come from a variety of backgrounds. I was classically trained as an ethologist and a neurobiologist and an evolutionary biologist. So I was not trained as a psychologist. So to me, we're bags of genes and chemicals, and how things develop in your brain becomes really, really important. And I often think that when we think of how do we stop puppies from being fearful, how do we stop cats from being terrified, how do we get horses to calmly walk with us instead of bucking, we're forgetting about the early stages of what happens in their brain, which is the big reason I wanted to focus on neurodevelopment. So what's neurodevelopment? This is the process of how your brain develops. And brain development is a process where connections between the cells in the brain, the billions and billions, and this is not an exaggeration, the current estimate for the human brain is there are 86 billion with a B, neurons, and how these neurons develop and form neurological pathways, because that is the key as to whether or not you have behavioral pathology. And when they develop these pathways are critical for your ability to do things like pay attention, to respond to social cues, to remember individuals and places and actions, and to make decisions and to have some control over the decisions that will keep you safe. That is all stuff that you are not born with and you have to develop. It's a dynamic process that's especially active in young puppies. And at first, very young animals of all species do the same thing. They just grow lots of neurons. And they've got lots of very disorganized, fairly long neurons that don't branch and touch anybody else because they have to start somewhere. And the whole point is just get your brain to function. That starts while you're still in utero, in the uterus. And it can be affected kindly and well favorably or adversely and poorly by what happened to your mother when she was a baby and what happened to your mother when she was pregnant with you. And that is across species. So the key is that you have to make these neurons, they're unorganized, you have to be able to start to organize them. And the way you grow them and the way you organize them depends on what happened to your mom throughout her whole life and when she was carrying you. The puppies mature and move through at first what we call neurobehavioral sensitive periods, which is what we're going to focus on today. And then they go through social maturity, which happens sometime between a year and two of age. And I'll close with a few thoughts about social maturity. During the time that you're developing these neurons intensely and at social maturity, they're intensely pruned and rearranged into more organized pathways so that the function is very specific to the brain region and to what we are now thinking of as nodal networks. These are not networks that are due to the region where they're derived. These are networks that are connected by function, and we can now measure these on MRI and functional MRI. And these networks are the things that appear to be impaired in many behavioral pathologies. So this organization that we go through depends on our ability to prune these. You maintain that ability at some level throughout your life. But as you move through your sensitive periods, and then again in social maturity, there's a big pulse. Transmission of information becomes faster because with pruning, new myelination occurs, and myelin is an insulator that speeds neurotransmission. And all of this is helped by what the puppies are exposed to. So when we compare these two neurons, and this is, let me just say this for people who don't look at these every day, this is your basic model cartoon of a neuron. And when you think that your brain has billions of these, and each of these neurons can make thousands of connections, sometimes tens of thousands, this is a very simple model. And yet this model has held up for over 100 years and it works. So when you look at the neuron, you've got pieces that come off it that are called dendrites that are very close to the nucleus. And then you've got a very long piece of it that goes a distance that's called the axon. And that axon also has branches. In this case, all you have is nervous tissue. There is nothing sheathing this. There is nothing insulating it. When you put myelin on here, which is a fatty acid that is derived from the supporting cells, the glial cells that help support the brain, what you end up by doing is you have fast neurotransmission, because now instead of going step by step by step by step, you skip between the nodes. So now you can communicate very effectively and very quickly. And part of social maturity, you're laying down this myelin, and then when you go through the early sensitive periods, you're creating it and remodeling it. And when you come back to social maturity, you lay down some more and you remodel it. So at both ends of that, this becomes important. Now, how behavior develops is really important. And I'm going to give you the schematic to show you where the sensitive periods fit in and to give you some context for some of the things I'm going to say. When you look at mom's summed environmental effects, this is everything she experienced in utero. This is what she experienced until she became pregnant with her puppies. This is what she is experiencing when she is pregnant. So pre and perinatal covers when she's nursing after she's given birth. All of those environments are summed. And all of those environments can have effects on the genome that she creates in each individual puppy. Thank you for spelling behavior with you. It's because it's a Canadian slide for one of my courses. Yes, you're welcome. I have feet and in both countries and passports for both, so it gets complicated. So what happens is when you create babies, you throw the genetic dice every single time. You, in theory, have half the genes from the mom and half the genes from the dad, and sometimes one or the other of those don't get read well, so the mother or the father has an overwhelming effect. But when you do this, you have a genetic code. That genetic code doesn't function simply by itself. Epigenetic effects are becoming more and better understood every day, and they are important. And nowhere, this is my personal belief, nowhere do I think there's a better place to look than in young dogs. Because young dogs undergo a series of environmental changes, and we have a lot of young dogs that experience trauma, and we can watch them grow up and do a series of tests that are non-invasive during their lifetime, and we can help them. And no matter how long I live, I'm not going to see a child born today through their entire life to see how well they'll do, but I can do that with a dog. So when we talk about epigenetic effects, if you take all of those summed effects of environments on mom, what ends up by happening is that you then look at the genome and you stick little methyl groups in places, or CH3 groups. And that means that if you have epigenetic effects, you may not read the entire genome. You may not have the heritability you think you do because you've either blocked the expression of certain genes or you've changed what they're going to be expressed as. So that's the genome that the puppy, and the puppy is all in blue here, that's the genome that the puppy is born with. It's the genetic part and the epigenetic part. As that puppy goes through the in utero environment, that puppy's genetic response surface continues to change based on whether mom had great pre-perinatal experiences or terrible ones. So mom could have had a terrible early exposure and still had epigenetic effects, but had great pregnancy and modulated the effect of those. When that dog is born as a result of those interactions, the way the dog behaves is what psychologists call temperament. That is thought to be relatively invariant through life, and it mostly has to do with how you arouse and how you react. It may not be invariant. As you go through the early exposure and sensitive periods in a series of environments, you'll get puppy and kitten juvenile behaviors by 6 months of age. By the time they get to 1 year of age, you're beginning to experience social maturity, neuronal pruning and remodeling, and you have your adult behaviors. And when we use scales or questionnaires that measure personality, that's the interaction of temperament with all of those environments across time and how you actually make your decisions and interact. Okay? Now, every time I have a little note about environments in green, you have the physical, behavioral, or social resource, which is nutrition and shelter and disease environments. Don't neglect these because a disease environment can affect all of this. So that's how you get to a behavior. And if that sounds complex, it's because it is. This is an example of a model for how these periods interact in humans. And one of the reasons we want you to do all of this synaptic remodeling and pruning your neurons and making better connections is if you do that, you are more plastic in your responses. Your brain can do more things. You can make more choices. You can sustain more trauma and damage. And in this graph, and this is out of the Harvard Center on Child Development, you can see that the yellow curve represents brain plasticity and influenced by experience, and the green curve represents the cost of modifying neurons so that you're less plastic. To modify neurons gives you plasticity, but there is a cost to that. You have to trim them. You have to make new protein. You have to make new insulators. You have to have all the resources to transport all of that in your brain. And it's not surprising that for the first 10 to 20 years of your life, the cost is very, very small in humans, and you're doing this in the extreme. As you get to the age of 40, this trade-off changes and it becomes more costly. Notice you still have plasticity by the time you're 80. The more you do, the more you keep. Okay, but as tissues age, the cost still goes up, even if you blunt that curve. Same thing happens in dogs. And when we are talking about socialization, I think that we have to realize it's a misnomer. It makes it sound like dogs' changes in how they develop and how they learn are restricted only to social situations and interactions with other dogs or other species. And when we say socialize, it also makes it sound like it's something we do to the dog. Not quite. We can help, but we can also hurt. So understanding neurodevelopment makes all the difference, because once you have a plastic brain, you're plastic about social situations, you're plastic about novelty, you're plastic about new environments, you're plastic about changing environments. And nowhere in the concept of socialization is there this nested concept of environments that may not be social, and they really matter. So let's talk about sensitive periods and neurobehavioral development. And the concept of a sensitive period takes the type of variation I was just describing into account and is best defined as a period when animals can best benefit from exposure to certain stimuli. And if they're deprived of that exposure, there's an increased risk for developing problems attendant with the stimulus. In other words, when animals are neurodevelopmentally able to respond to the stimulus, they'll benefit from exposure. And if they lack exposure, they could develop behavioral problems associated with that omission. So not all of us develop at the same rate. If we all had access to phenomenal resources, we all still wouldn't develop at the same rate, but we would cluster differently than what happens when you've got deprivation or trauma to mom. And when we look at the core of these sensitive periods together, this is often labeled the critical period for social and environmental exposure. And when we look at this in humans, and there are papers on sensitive periods in humans, and this is a recent one. This is a 2020 paper in Current Opinions in Behavioral Sciences. Sensitive periods involve experience expectant learning processes that shape the development of sensation and perception. In other words, how you get through your environment, sight, sound, touch, and perception, what that means to you, how you perceive those things in the context of everything that's going around, as well as affective, which means emotional, and cognitive processes. Sensitive periods shape how you're going to respond to emotional and cognitive events for the rest of your life. These matter. When appropriately protected, challenged, and exposed during sensitive periods, such exposure promotes the plasticity we've been talking about, neurobehavioral plasticity, which allows you to recover and learn new ways of traveling through life. At the molecular and neurological levels, you have more and better neuronal responses that promote the ability to survive and thrive. And that's the whole point. This is an evolutionary biology argument. The evolutionary biologist in me is telling you that the reason you want to do this is the whole point of the brain is to be able to make sure you survive and, in theory, pass on your genes, but that you can get through and navigate a world that is not always a kind and safe place. And I think that's what we all want. And if you can do that, my goal in life for my patients is that they have joy. So they have to be plastic. They have to get over their fears to be able to have joy. So when we talk about brain development, and this is yet another human example, and I'm giving you this simply because it's easier to understand when you've got 9 months of gestation rather than 2. So you begin to develop the nervous system itself as in the embryo within the first month of pregnancy. You begin to grow new neurons for the first 3 months, and then they begin to move. So they migrate through different levels of the brain, but they're still not this sophisticated, interconnected, pruned, insulated network that we're talking about. That doesn't happen for a while later. That starts to happen at the very end of pregnancy and then continues well into adulthood. Notice they don't have it stopping here because it doesn't. It's the rate of speed that changes. You then begin to make, when you're at about week 18 or 20, you begin to make these synapses. And as soon as you talk to other neurons, now you begin to prune them and to remodel things because you're beginning to see what adaptations and what changes need to occur. That occurs certainly through adolescence, and you're going to start to prune them. And that occurs before you even make the synapses. Some of these neurons are just in the wrong place, or they're not any good, or they're not doing anything, and they go away. And you can go through pruning them well through childhood. And then as you go through adolescence into adulthood, this pops up again. So when we're talking about puppies, pretty much everything that we know about puppies, and I am appalled by this, was published by Scott and Fuller in the 1950s and 60s. And I'm not knocking Scott and Fuller. I was the speaker for J. Paul Scott's memorial years ago. And I remember saying, if he had at his disposal, the tools I have at mine, my career would be completely redundant. I mean, the man was brilliant, but you can only do what you have the resources to do in your lifetime. And as somebody who has spent their entire life doing research and designing research studies, I can tell you this isn't easy. It takes a team, it takes a community, and it takes all the help anybody can get. But what we know from the limited studies that his group did in a rat lab, by the way, at Bar Harbor, on 5 breeds of dogs where they bred a number of litters of 5 breeds, all of whom were the same size and shape. So they had beagles and fox terriers and Basenjis. You'll see some of the data in a minute. The point wasn't to cover every breed group. The point wasn't to cover all the breeds. It was to control for shape and size because of the way they had to house the dogs. That's the truth. The neonatal period was birth to 2 weeks. The transitional period was 2 to 3 weeks. Now, neonatal period, the thing I did not tell you when we talked about humans is when you were born as a human, you can see and hear. You don't do a great job of it. You don't see all that well. When you're born as a puppy, you don't see and hear. So that's what this neonatal period of the first 2 weeks is, where you're further maturing neurons that are already mature in human babies when they're born. And by the way, in giraffes when they're born. Okay. So not all species are the same. The transitional period where your brain is maturing enough that it can begin to undergo social exposure and have it be a meaningful experience is 2 to 3 weeks. By 3 to 5 weeks, you're in one of the exposure periods that we're going to talk about. By 5 to 8 weeks, you've moved into another. And by 8 to 12 weeks, you've moved into a third. And those 3 periods of time together are called the early exposure periods. From weeks 12 to 6 months, they've been broadly characterized as the later exposure periods. And from 6 to 12 months, we consider a dog to be a juvenile or in adolescence. And then by 12 to 18 to 24 months of age, they're undergoing this period of social maturity. So what happens in canine brain development? This is a set of 3 or 4 slides that just is going to take you day by day, week by week, through the first 16 weeks, 4 months of a dog's life. So you're maturing the cranial nerves from days 0 to 13. That's the neonatal period. And unless your cranial nerves the ones that feed all of your um, reflexes in your head, so your ability to blink, your ability to twitch your nostril, your ability to move your eyes in different directions, to hear, to swallow, to have sensation. Those aren't mature when you're born. They mature in the first 2 weeks. You begin to myelinate the somatosensory and motor cortex by 3 weeks. Dogs raised with a species that's not their own, and it's usually cats in these studies, and there are only a couple of studies, from 13 days through 13 weeks, don't ever recognize themselves as dogs and don't recognize dogs as being the same as them. So that tells you that this period is essential for forming your identity. They're able to begin to identify other species from 13 days through 3 weeks. That's when it starts. The startle reflex, once you've got your cranial nerves, you can startle. So 13 to 20 days, response to auditory stimuli the same, and response to visual stimuli the same. Your teeth emerge at about 3 weeks. Starting at about 3 weeks, you engage in rough and tumble play that becomes the modus operandi if you're a puppy through 8 weeks of age. And after that, it will taper off if you're not stimulated. Tail wagging begins starting at 3 weeks through 8 weeks. And I'm going to show you Scott and Fuller's original data because this is when it begins and there is range. Increased locomotor ability kicks in at about 3 weeks, as does the ability to explore other dogs, approach new people, follow each other, and eat semi-solid food. All of those things start. They're not over. They start at 3 weeks. So think about it. Now you can get up, you can get around, you can begin to explore different foods. You can follow each other because you can move well, you're a little coordinated. Oh, and look, you're recognizing different species. You're able to meet new dogs and new people, and that's going to continue. By 3 weeks, you begin a rapid myelination period of the somatosensory cortex. By 4 weeks, you can begin to bark, and there's a range of ages for when you start, but it's 4 to 7 weeks. By 5 to 6 weeks, you'll mount your litter mates, and by 5 weeks, this is when you see maximum distress calls in puppies when they're unsure of something, and it's 5 weeks of age. And for that reason, that was originally thought to be a marker for a part of one of these periods. You hone interest-specific skills at the same time, so it's not surprising you might get a little upset from time to time, and by now you are switching to eating solid food. In dogs genetically bred to be fearful, the first pathological responses occur right at about 5 weeks. There are some few dogs at 4 weeks and some dogs by 6 weeks, but having tested a number of these dogs in large colonies, I can tell you 5 weeks, you're going to pick up 90% of them. Dams naturally wean, but will stay with the pups through 10 weeks of age, starting at 7 weeks. So if you don't separate mom, she's automatically going to wean them at 7 weeks, and she'll still stay with them. She might occasionally let them nurse, but it's not nutritive to any great extent by 10 weeks of age. You're going to continue to really myelinate the cortex from 5 through 8 weeks, and that's when you're going to see the first signs of an adult EEG. And house training is most successful at the end of that period because finally you have a cortex that is myelinated, that is thoughtful, that can make associations. So you can make the cognitive association between a good place to eliminate, and you have the neuromuscular control to inhibit elimination. Play behaviors in free-ranging or homeless dogs decrease between 9 to 10 weeks, which is really young. But that's also the time that the rough and tumble play will decrease unless you stimulate it. If you're not exposed to new environments by 14 weeks of age, not at, by 14 weeks of age, you'll never explore them. And major myelination gets completed by about 5 months. So through 20 weeks, you're going to complete it, and it gears up starting at about 14 weeks and goes from 14 to 20 weeks. And you've really myelinated everything in the dog brain. You're going to continue to prune and remodel the brain through 20 weeks. And this is the first time you'll actually see marking behaviors begin to appear as dogs approach sexual maturity, which for males is 6 months of age. Now, here's the Scott and Fuller data that I said I would show you, and I'm going to show you one piece of it. It's an amazing array of data, but I just want to talk to you about tail wagging because there's an evolutionary argument here. The problem with breeds, and breeds you should think of as pools of canalized genetic variation, they vary. So we may not know what the range of normal is. It's shocking to me now, and it has been shocking to me for the 30 years of my career, that no one has kept a database on when puppies begin to wag their tails. This is a very important behavior. It's something any breeder could mark down. We now have the ability to do cloud-based research. Why the AKC isn't collecting half the data it should, I have no answers for these things. But I've got a million research projects percolating in my brain that would help us understand normal. And this one is simple. So here are their 5 breeds: Basenjis, Beagles, Cocker Spaniels, Shelties, and Fox Terriers. Number of litters they had, the number of animals that they had total, so 160 total animals. And they separated them by male and female because they're smart. If you lump everything, you can never figure out who is a male and female. But if you count the males and count the females and measure them separately, you can always lump them later, but now you have sex information, and it's valuable here. So if you look at the first day this was observed, the last day that a dog in the litter began to wag its tail, and the median number of days, you're not going to be impressed very much by the median number of days. But look at the first day. So who was first? We have 2 breeds, Cockers and Shelties, that had dogs wag tails at 17 days of age. Interesting. Who was last? Basenjis. Here we've got Cockers and Shelties asked to be very interactive with humans, to use their tail in a lot of situations. Here we've got an ancestral breed. Do you think people read those breeds as they are growing up as puppies in the same way? And I want you to think about that, because if you're trying to make your puppy be everything it can be and keep it safe and happy with a plastic brain, you have to understand what it's signaling. And if you're relying only on tails, we've got a real problem. And this is an example of the type of situation where people get into where they don't even realize they begin to treat animals differently because of what they don't see or what they don't know or what they don't understand. And it may be important. So why are these sensitive periods for social and environmental exposure so important? This is all about risk. Everything I think about is about risk. We lack good discrete data across types of dogs, whether it's breeds, sizes, lines. So we need to err on the side of caution to ensure puppies get what they need. Neurodevelopmental and neurobehavioral risk is real for poor prenatal, maternal care, and nutrition. So those are the epigenetic effects. Lack of exposure, hyperreactivity, hyperarousal, fear, and panic. Too much imposed exposure or imposed exposure of the wrong type, fear and panic. I can't tell you how many dogs we see in the clinic where people said, well, she was a little shy as a puppy, so we kept taking her to dog parks and exposing her. Yeah, now she's basket case. That was really good. How much plasticity can we build back into that dog? If they're under 2 years of age, we have more space than if they're 5, because they're going to remodel their brain. This is a tabular version of that graph I showed you. So I'm just going to go through the periods of days. I'm going to talk about what problems occur if you miss the stimuli during those periods. And you can read about the things that would help the most. But so 0 to 13 days. This is the period of time where your eyes and ears aren't really open. If you're not handled and you don't get some exposure to vibrations that isn't scary, believe it or not, you have altered sensitivity to touch and you are hyper reactive to anybody who touches you or makes certain noises. The startle reflex, when you can hear and you can see, if you miss exposure to sounds and sights that are different and challenge you a little bit without scaring you, you may have concerns with visual and auditory acuity. And if you're absolutely raised in a depauperate environment, you may not have the range of abilities that other animals would have. From 3 to 8 weeks, you're beginning to be exposed. This is the time period where you're beginning to follow around other animals. You can get up, you can get around. You've got that 5-week period in there where you're suddenly recognizing new humans and new dogs, and you realize, oh my God, things could go wrong, and you scream and you get scared, and you suddenly realize the world can fight back. If you miss the chance to have that exposure, you have heightened reactivity to dogs, heightened reactivity to other species, including humans, lack of inhibition for your arousal levels and behavioral responses, because you can learn to calm and settle or relax. And that learning has profound responses for how you later handle situations that are potentially anxiety provoking. Again, when you talk about the fear of these other things, what's happened is you've got a lack of learned inhibition for loads of things, but key here is for elimination. If no one began to expose you to an appropriate elimination environment by 7 weeks of age and reward you. I would start puppies out at 5 weeks of age. You don't know what the tail ends of the distributions are. And every time they piddle or poo in an appropriate space, tell them they're brilliant. There is no role for punishment here. But you have to be able to cognitively make the association that this is good, this isn't good, and you have to be able neurologically to inhibit micturition. Now, why do I make such a big deal of that when we're talking about fear of other dogs and fear of species and fear of approaches of humans, all of which happens in the same period? Because the first time somebody abuses an animal is when they're house training. And if they understood this, they would stop doing it. Most of them. Okay. The earliest age at which a dog should be adopted is 8.5 weeks for this reason. Give them the best start. From 14 to 20 weeks, if you miss those exposures to new environments, you have profound panic. The plasticity of the response of normal behaviors is gone, and you can't respond with a variety of choices. What is so classic for pathological or abnormal behavior is that they're not plastic. If you watch a video of a dog doing something tremendously abnormal and you play it and fast forward, you'll notice the episodes of the abnormal responses are very stereotypic. They just pop right out because they aren't able to have a plastic response. They aren't able to have a measured response. They aren't able to say, well, if not A, maybe B. If not B, maybe C. The ability to have a plastic and flexible response and to have neurons that facilitate that is learned, and it's learned behaviorally, and it's entrained and learned at the neuromolecular level. So if we know all of this and there's risk, why don't we just get on with it? Well, people may not actually know this. Many people think dogs and children raise themselves. People don't understand that puppies can be abnormal. People have a tendency to think more is more when less is more. Think about the dog park. Finally, even when they do get it, people worry about immune systems of young pups and attempt to protect them from communicable diseases. And when they do that, they are at high risk to do profound neurobiological damage. This is an illustration of what's called the immunity gap. Here are the maternal antibodies that come from colostrum and they begin to drop. This is week 4, this is week 12. Here are the critical periods. So you've got these dropping beginning at week 4. By the time you get to week 6 to 8, they're really dropping. And by the time you get to week 12, they're low. Antibodies produced by the puppy or kitten, this is the lower antibody threshold that you want. This is the lower inhibitory threshold of the vaccine response, begin to come up. And if you boost these with vaccines, you're beginning to develop a level of immunity that looks like an adult. If you add these two curves together, you will get to this response. But to do that, you've got to have a healthy puppy going in who has an immunoresponsive system, and you have to vaccinate, and they have to have excellent sanitation and sensible exposure. Why does early exposure and development matter so much? Well, it matters because of deprivation. And I'm going to give you an example of why how mom is treated matters. And this is the pre and postnatal deprivation story. And it's the story of what happens when you have too much cortisol. So what we're going to be talking about here are how epigenetic effects, and mom herself may have experienced epigenetic effects, but how mom's pre and perinatal experiences can affect what happens to the genome. And I'm going to give you an example. Epigenetic effects are what happens when glucocorticoids, cortisol, are in excess pre, peri, and postnatally. They affect what happens at the cellular and molecular level. Chronic glucocorticoid exposure affects the structural development of the hippocampus, the part of the brain involved in associational learning. Sit, stay, good girl. Chronic cortisol elevation actually interferes with the acquisition and consolidation of task learning in certain regions of the hippocampus. And prenatal and ongoing stress in the rat model leads to lower levels of extinction of cue-conditioned fear, like a fear of learned noises. Okay, cue-conditioned fear. So certain noises trigger you to have a specific response because you've shrunken the hippocampus. You shrink the hippocampus and you have subsequent memory impairment. When you shrink the hippocampus, the amygdala is actually more reactive. So the part of the brain involved in fear is more reactive. And you facilitate that, especially for auditory fear conditioning, because it's a very specific case of cells that are hyperreactive to certain stimuli. And these effects are worse in some genetic backgrounds. So if you have a puppy who comes from a line of dogs where you've got genes involved in noise reactivity, you can have these types of environments. And yes, animals living in labs can have unnatural environments, but they don't have to. And when you take a look at a well-run lab, it's a very protected environment. And then when you add in some of these noises and stimuli and stressors, you see the true effects that can happen. Whereas if you didn't start out with a protected environment, which is what most puppies don't start out with, you're going to have a more pronounced effect in puppies. So when we look at maternal grooming behavior in mice, there are genetically great mothers. I've switched, by the way, to mice here. There are genetically great mothers and there are genetically lousy mothers. These are genetic lines that have been selected for. The lousy moms don't groom. The great mothers do a lot of grooming. The great mothers are used as the model for you're well-fed, you're getting the care, you have very few epigenetic effects. The low grooming line is used as the model for high epigenetic effects. When you look at these types of things, you're looking at the gene that is going to be involved in producing glucocorticoid receptors. You need glucocorticoid receptors because they're going to suck up any cortisol that whenever you get upset, you are going to release. So what happens here is these are the methyl groups I was talking about. These are CH3 groups. Here's the promoter region of the gene. Here's the gene for the glucocorticoid receptor, and you have a couple of methyl groups here, but you can still transcribe this gene and you can go on to make lots of glucocorticoid receptors. In the low maternal grooming line, you have a lot of methylation. You have a lot of epigenetic effects. Now you can't take the promoter region factor and get it to stimulate the promoter region. They need to lock to get that promoter stimulation. As a result, the promoter region doesn't transcribe, doesn't tell the gene to go on to transcribe a lot of glucocorticoid receptors, so you only get a few in the hippocampus. Now what happens later? So now you've got the pups, they're at the same age, and they're subjected to some type of stress. Maybe it got cold, maybe a new rodent was introduced. And they put out glucocorticoids, they put out cortisol, and because their hippocampus has normal glucocorticoid receptor expression, they can inhibit that stress response. And they're like, oh, it was just another mouse going by. Oh, it just got cold. In the line that has epigenetic effects, they have the same stress response, but they can't inhibit it because they don't have enough receptors to suck it up. So they're always, always, always stressed. And that's what's happening in so many of these puppies. They are being stressed chronically. And furthermore, they may be being deprived of food because mom didn't have food. And once they're deprived of food, they stimulate a hormone that stimulates neuropeptide Y, and now they kick in two of the stress pathways. They just don't go through the regular stress pathway, but they go through the arginine vasopressin pathway. So this is the type of stuff that puppies undergo all the time. And I get that some people don't like lab research. And I saw the comments about animal care committees. And let me tell you, I have overseen animal care committees. And when this is done well, it's done brilliantly well. But there is no animal care committee for these puppies that you're seeing. And there is no animal care committee for the pregnant bitches brought into shelters. And these animals are undergoing the same stressor response that the rodents are. And we only understand this because of the rodents. And this is not a humane way to raise a mother or puppies. And the only way a pup can begin to recover from this type of trauma, if they're born this way, is to ensure those sensitive periods are really exploited to the max. And by that, I don't mean you expose them to everything in sight. You make sure that they go at the pace they're able to, and that they do the best they can, and that they can develop the most plastic response possible. Because remember I said it's got to be plasticity at the behavioral level plus at the neuromolecular level. This is what happens when you learn something. You have environmental inputs. They can be toxins or viruses or psychological drugs. You can stimulate proteins, neurotransmitters, ion channels, receptors, loads of things. Those things all then move into the nucleus of the cell and tell the genotype that you need to make new proteins. The only way you learn something and remember it is to make proteins. So you have to stimulate that promoter region, and then you have to turn on the coding region. And then you're going to make more neurotransmitters and more receptors and more ion channels and more cell membranes and loads of things. And that's what gives you behavior. If you can't do this, you can't learn. And what sensitive periods are about is learning to learn at both the behavioral and the molecular levels. And we see impaired rescue puppies that have limited ability to learn until they've received treatment. This is an example of two of these dogs. These dogs are 3 years old. They are litter mates. They were shipped up from Texas to the Atlantic provinces, and they are terrified of everybody except their people. That's them at their first appointment. That is not how they are now. But you can see that this dog thinks everything in the world is going to come for him. That is not a great way to grow up. And we have some other data from dogs. This is a paper that looks at the age of separation of two groups of 70 dogs each. One of them was separated by 4.5 to 5.5 weeks of age, and the other was separated at 8 weeks of age, which is the recommended age. And they found out that the dogs that were separated earlier statistically significantly sought more attention. They were more reactive to noises. And the other behaviors that showed up enough to be a concern, but were not significant in a sample size of 140 dogs, was excessive barking, aversion to strangers, destructiveness. My point is, you can see the pattern here. Attention-seeking behavior and reactivity to noises is enough of itself to be its own hell for a puppy. So separating pups early interferes with those sensitive periods because you get that stress response, and now you can't learn, and you've affected the hippocampus adversely. It shrinks, and you've got a hyperreactive amygdala, and you become more afraid. And this is some of the strongest evidence that we shouldn't let pups go before 8.5 weeks, or 8 weeks at the minimum. I'd prefer 8.5. This is the story of one of our dogs. This is my miniature Aussie, a breed I swore I would absolutely never have. But this dog was one of our patients. She was scheduled for her recheck for truly neophobic responses and pathological fear at 11 months of age. And a week before the appointment, she'd been doing well according to all the reports that we had by our Zoom calls. A week before the appointment, the client showed up and made an appointment for euthanasia through the emergency service, and I had 15 minutes to find this puppy a new home. So it came home with me. And this is the most fearful dog I'd ever seen. The woman was at the end of her tether because this dog was not completely house trained, and the husband wanted the dog out of the house. And the dog became more and more fearful and wouldn't eat, and without eating, couldn't take her pills and completely regressed. And her whole story of neophobia was simply that this is a puppy mill dog from Quebec. Apparently, she was injured at 2 weeks of age. I've never seen any evidence of the groin injury, but they took her away from everybody else and raised her by herself till she was 8 weeks of age. Terrible idea. And she was afraid of everything. So this is the first day at our house where she was able to go outside on a lead. That occurred 3 to 4 weeks after she came into our household. It took her over a week to be able to get from the garage to the breezeway to a bedroom. And the dog who is helping her with this is Gabriel. And Gabriel became this dog's gateway to sanity. And she is now absolutely delightful, nearly 2 years later. So what about source of dog? Well, in Italy, there are not puppy mills, but you could take your family bred puppies and sell them in a pet store if you don't have a home. So this was a survey study, and survey studies have their own particular sets of problems, but they were able to get quite a decent sample size, and they simply asked people, did you get your dog from the breeder? Did you get it from a pet store? And when they looked, dogs who came from pet stores had a greater incidence of body licking, house soiling, owner-directed aggression of any kind, and separation-related behaviors. So at least twice the amount of separation-related behavior, 3 times the amount of house soiling, and 2.4 times the amount of aggression to any human than the dogs who were just simply raised in households. So the difference in narrowing a dog's world and closing off that exposure is a problem. So what can and should we do? We need to care for the mom. There needs to be an excellent plan of nutrition, high DHA and EPA, the omega-3 fatty acids, the polyunsaturated fatty acids, an excellent vaccine program. We need to ensure that during the sensitive period, pups have access to the relevant foci for exposures so that they can use that exposure. They need to have excellent puppy classes. This is Gabriel when he was a puppy. Yes, he is an Australian Shepherd. Yes, he does have the genetic defect for coat color. Yes, he is bald now as an adult. So he's gray, and he went on to lose most of that very funky hair. But he was a hit at puppy class. He loved the balls. He got on a skateboard. They had breaks outside where they got chew sticks. He was exposed to all sorts of animals, and animals that are known to you are great teachers. So here is Hamilton, who was 18 months older than Annie and raised Annie. And here's Annie with baby Gabriel, and she's 18 months older than Gabriel, and she's raising Gabriel. And she raised him through all of his puppyhood stages. And Hamilton kicked in. And yes, we let our dogs stand on marble tables because, you know, that table's a little wobbly and slick, and it's a good novel surface for him to learn about. And Annie is a great teacher when puppies get rough because she doesn't take a lot of grief. And you want to incorporate into daily life a way that they feel safe and can have redress bit by bit, things that might be a little scary to them, new things. So Gabriel was 4 months of age when I put him in a swim vest, and I stuffed him inside my emergency life vest, and I took him in my kayak with me. And he did get a dunking in the water and found out he could swim, and he now loves to swim in the water and loves to swim with the kayak. He no longer fits in my sea kayak. If pups seem reticent to undergo exposure, give them a bit of time and protect them. But if this is continuous, if it happens after a week or two, get help. What was the thing that made the big difference in Ladybug's life? Medication. This is the most fearful dog I've seen in 25 years of treating patients. This dog is still on a cocktail of medications that would shock most people. She started at 6 months of age, and if she hadn't had that month and a half where she couldn't have taken her pills, she would have done brilliantly. And as I said, now, yeah, she had a supportive place. She had a big space. You have to realize this dog couldn't go out in this big space at first. She couldn't approach males. She couldn't approach new things. She now lives on the beach. She has a dog who helped her. It made all the difference that she had the medical care through a specialist, me, that she needed. The last thing I want to mention is social maturity. And I know we're coming to the end, and I'll make sure we take some questions. The material you have to work with in social maturity is determined by the earlier effects. What happens in these social development periods? What happens in these sensitive periods? What happens with the environmental exposure? That's that juvenile dog that you've got in that graphic I showed you. This is the stuff that you now have to remodel when you're going to, again, develop new neurons, prune them, and remyelinate the brain. And in humans, we know social maturity is a period of somewhat renewed but progressive myelination and regressive pruning. So you're going to trim old neurons. That's associated with changes in neurochemical profiles that you can see on imaging and shifts in behavior. And we've not yet measured this in cats and dogs. I currently have a grant pending again for that. And I think it's critical that we do this at some point. And before I retire, I would like to have this knocked. As humans age from 6 to 17 years of age, cerebral gray matter volume decreases, and white matter, which is the myelin, increases. And that helps you to integrate and act on information. Standard MRI assays reveal profound differences in size by age trajectories for males and females as they mature through 27 years, and connections between frontal and temporal lobes develop more slowly than other regions. We have no idea if this is true in dogs. Change in maturation of the human frontal cortex improves cognitive processing. These brains are homologous. That also should happen in dogs. But if they were traumatized early and then didn't get the exposure and the enhancement they needed at social maturity, you're not going to improve their cognitive processing, which is why all of our behavior modification is focused on enhancing their cognitive performance and teaching them to decrease their stress levels, decrease their glucocorticoid loads to do so. We know in humans that trauma affects maturation, and we know this through imaging studies. And we can hypothesize that the same pattern occurs in dogs and cats. In dogs, You can begin to see these changes as early as 10 months, and the dog that you suddenly thought was the most brilliant puppy you've ever had left its brain in the back pasture. And the good news is 4 to 6 months later, it's going to go back to the back pasture and find its brain. But while you're undergoing this remodeling of your brain, you're not the dog you were before or the dog you're going to be afterwards. So there's a range of 12 to 36 months that people cite. I tend to think it's going to be 10 to 24 if we ever get the imaging studies done. But we should expect the same types of data we see in humans. So let me just summarize the key points. How prepared you are for social exposure depends on the pre, peri, and postnatal effects of mom. And I know it's upsetting to think of deprivation, but it's happening every day in every species. How do we mitigate this? We're going to use the omega-3s, the DHA and the EPA, and protect pups from things that scare them. Early exposure is indicated by stage and focus of neurodevelopment, and it's essential. How are we going to mitigate this? Expose gently across context to the level of the dog's comfort. And we're going to engage in a state-of-the-art core vaccination program and know what other vaccines are recommended where you live and vaccinate. And the mitigation is cleanliness and an emphasis on biosafety. Great puppy and early training classes. Mitigation, assessment by veterinarian or behavior specialist, medication one-on-one with a certified positive reinforcement only trainer. Every puppy class should have one person for every 2 puppies. That's what my dog's classes have had. And every effort needs to be made to protect neuronal growth and plasticity. And I talk to my clients this way. When we see puppies, this is what I explain to them. Everything I just explained to you, because I want them to understand that that's not just a dog in front of them. That's a dog's brain. And we need to make it safe and make it everything it can be. So I'm going to stop there. Anybody can reach me. This is my UPI email address. This is my private email address. For those of you who are veterinarians or veterinary nurses, we offer a 1-week intensive course every year called Behavior at the Beach. It's full this year, but next year it's going to be the 31st of May to the 5th of June. Announcements will be out, but you're always allowed to reach out and find out exactly when. Prices are going to be comparable to this year. They're not going to go up that much. For those of you who might be interested in working dogs, there's a conference in another 6 weeks in Warwick, UK, about service and military and police and working dogs. And I'm going to give some of the data from our early puppy study. And I edit the journal in the field. And there's my poor rescue border collie who, while we were setting this up, decided as soon as he saw Sheila, he had to start barking. I'm going to stop and I will be pleased to answer any questions that are still out there.
Wonderful.
I've upset.
Thank you so much, Dr. Overall, for this fascinating presentation. And thank you to everyone for joining us today.
Well, thank you.
So